this experiment already during the last practical, which is on acclimatization of your tissue cultured plantlets. So if you can see in this picture here, nampak kan the picture on the front slide?
Nampak doktor.
Alright, so as you can see here,
Nampak.
basically what you've done in the during the last practical, where once you have successfully produced your tissue culture plantlet, the next thing that you should do is you have to induce production of roots. So you transfer your shoots into a suitable rooting media, and once they have successfully produced adequate amount of roots, then you can take them out from the in vitro environment. In vitro is when you culture the plants inside the culture vessel. So that's when we call that the condition is in vitro. But once you take them out outside of the culture environment, then we call it ex vitro. So then for acclimatization, you will transfer your plantlets ex vitro into a suitable growth media, usually composed of fertile soil lah, that is usually autoclave. Autoclave means macam you buat your media, you put your soil in an autoclave machine. The reason why you do this is you want to sterilize the soil so that you get rid of microbes, right? Why you want to get rid of microbes? This is because when you do tissue culture, you are basically growing your plantlets in a condition where they are free from any microorganism, right? So that's why the soil that you grow your plantlets in must be in the same condition, must be free from microorganism, or else then the plants may possibly die because they are not used to resources competition, right, with the microbes. And then as you can see in this picture here, they are covering the top part of the pots with the plastic macam container. Sama lah like last time you cover your pots with apa a piece of plastic, right? It's the same thing. It's to maintain the high humidity level for your plants. Right. Okay, more people joining. Okay, I think that's about it lah for audit 26. Okay, moving on. Okay, so why do we need to acclimatize? Why is the need for acclimatization? Right? So from what we've learned before, we know that when you are doing plant tissue culture, it means that the growth and the multiplication of cells, the production of tissues, the production of organs of your plants are actually done on either solid or liquid media, depending on the media composition that you use, and they are all done under aseptic conditions. Aseptic means free from microorganisms, very clean environment, and the environment is controlled, meaning that you control everything. You control the exposure to light, the amount of nutrients that you supply to your plants. Right, so you control everything. Okay, so your plants don't need to do or don't need to exert any effort at all in order for them to continue growing, right? And then because of this, there is also a major limitation. Okay, because when you do plant tissue culture, you are basically producing all of these plants in the lab. So the result that you will get is high, right? Because you are multiplying uh, the production of your plants in the lab under controlled conditions. So from one explant or from one mother plant, you will get like hundreds or thousands of subsequent cultures. But there is one limitation for this large scale application of tissue culture is that there will be high mortality after you transfer your plants from the lab to the land. Okay, which is the main or the biggest hurdle that any tissue culturist would face, which is in the lab everything is going on so smoothly. You get a lot of micropropagated plants, but these micropropagated plants are not of any use to you, right? Because the reason why, sorry, because okay, I think this is the last one. Okay, the reason why is because um you get a lot of these cultures in the lab, right? But the reason why you want to do this tissue culture protocol in the first place is so that you can mass produce plants of high yield, or maybe mass produce ornamental plants that has very high value. So the only thing that would prevent you from profiting from this technology is the fact that you cannot transfer your plants from the lab to the land. Right, you get what I mean, right? Because you need to transfer your plants to the land so that they can continuously grow like no wild type plants as well, right? And why? or what are the causes of this high mortality rate that you observe when you transfer your plants from the lab to the land? This is because the microshoots that you produce in vitro, when they are transferred ex vitro, you are exposing them to abiotic conditions. Abiotic, there are two types of stresses or two types of conditions. We have biotic as well as abiotic. Abiotic means anything that is not of biological origin. Biotic means anything that is of biological origin. So when you are transferring your plants from in vitro to ex vitro, you are exposing them to a lot of these abiotic stresses. For example, altered temperature, whereby in the environment the temperature will be fluctuating, right? Sometimes very cold, sometimes moderate, sometimes very hot, and the light intensity also would depend on the exposure to sunlight, and then the humidity as well will vary, unlike the conditions that they are used to in culture, right? And other than that, these plants will also be exposed to biotic stresses or biotic conditions. For example, soil microflora, which is the soil microorganism. So in order for you to make sure that your plants can tahan or can withstand all of these stressful conditions, you must acclimatize your plantlets. You must get your plants to be used to this condition first before you can successfully transfer them to the field or to the land where you are growing your plants at, right? This is to ensure for successful establishment as well as survival of the plantlets. There are two keywords here: establishment and survival. Survival is to survive. Establishment means they can grow and continuously grow, right? And in order to increase growth and reduce mortality in the plantlets during acclimatization, a lot of research has been done to focus on how to control the environmental conditions. For example, physically, chemically, and then to acclimatize the plants so that they can compete with the microorganisms that are commonly found in the soil. Alright, so there are various factors that will affect the success of ex vitro acclimatization. First is induced altered morphology and physiology. So you know that when you grow plants in vitro, obviously they will be very different from field grown plants. Right? For example, if you look at the figure on the right here, this show you tissue culture produced plantlets with some roots generated. So these tissue culture derived plants will show induced altered morphology and physiology, meaning that the morphology of the plants as well as the physiology of the in vitro plants have been developed so that they are accustomed to the growth environments that you provide for them in culture, right? And the cause of high mortality upon the transfer to ex vitro environment is mostly because these cultured plants, they have non-functional stomata, weak root system, and poorly developed cuticle. So why is this? This is because in tissue culture plants, obviously you are providing your plants with nutrients, sufficient amount of light, and high humidity level, right? So tissue culture plants will have very weak stomata because you are actually limiting the need of the plants to conduct photosynthesis, because everything is you're going to provide for your plant, the nutrient, etc., right? And then you also have weak root system because again the nutrients are supplied to the plant, so they have weak root system because they don't really have the need for very efficient nutrient uptake, right? And then they also have poorly developed cuticle. Why? Because again, they are not exposed to very high temperature, so they don't really need that thick cuticle to prevent from excessive dehydration or transpiration. Right, and then in the culture as well, you are supplying your plants with excess or a lot of phytohormones. Therefore, the plantlets will show abnormalities in terms of their morphology and anatomy. These are called vitrified plants or hyperhydric plants, like we've learned before, or plants showing symptoms of vitrification, as shown in figure B here, compared to the wild type plant. Sorry, not the wild type, compared to morphologically normal plant in A. So B shows you the vitrified or hyperhydric plants, and the physiology and the anatomical characteristic of these plants will necessitate that they should be gradually acclimatized to the environment. Gradually means you are slowly exposing your plants to the conditions that they would be exposed to in the outside environment. So you have to do this gradually. That's why in your practical previously, you are covering your pots with the plastic, right? This is to ensure that the humidity level for the plants will remain high. And then as time goes by, we will slowly increase or maybe make some opening on the plastic so that they will be exposed to outside air. So in many plant species, the leaves formed in culture will be not able to develop further after ex vitro condition. And after this acclimatization, they will be replaced by newly formed leaves. Okay, why is this? This is because these in vitro grown plantlets will have leaves with large stomata with a different shape and structure. For example, the gut cells will have thinner cell walls and will contain more starch and chloroplast, right? And then in a study by Ticha et al. In year 1999, they found that the acclimatization of tobacco plantlets to ex vitro conditions will decrease the stomatal density. Stomatal density means it will decrease the number of stomata per unit area of the leaf. And the size and the morphology of the stomata will also be changed on both sides of the newly formed leaves. This is again to ensure for the plants to be able to survive in the outside environment. So during acclimatization, various morphological changes will occur. For example, leaf thickness will increase. The leaf mesophyll will will differentiate into palisade and spongy parenchyma. And then again, I've mentioned before, stomatal density will decrease. Why is there a need for the plants to decrease their stomatal density? It's again to reduce transpiration because they are not used to this hot environment outside, right? So they have to reduce their stomatal density so that they can maintain efficient photosynthesis, but at the same time reduce the amount of water that will leave the plants through transpiration, right? And then the stomata form will also change from circular shaped stomata to elliptical shaped stomata. And then there will be development of cuticle and epicuticular waxes. This is to allow for more effective regulation of transpiration through stomata, right? And then to stabilize water potential of the field transferred plantlets. Okay, let's talk about abiotic stresses. So again, I've mentioned before, when you transfer these plantlets into outside environment, they are exposed to biotic and abiotic. Abiotic means anything that is not of biological origin. So first is light intensity. So when you are doing tissue culture experiments, you are basically maintaining your tissue culture plantlets in the culture room, right? And these plants are kept under low light intensity from 1200 to 3000 lux only. And then the temperature you also control in the culture room. You maintain your culture room at 25 plus minus 2 degrees Celsius, for example. Therefore, if you directly transfer your plantlets into the environment, right, you are actually transferring them into a broad spectrum sunlight, which has very high light intensity, which is not suitable for your plants, right? So the light intensity in the outside environment is from 4,000 to 12,000 lux, depending on the location, right? And temperature also varies from 26 to 36 degrees Celsius. So these conditions, this exposure to high sunlight as well as high temperature may cause charring of the leaves and the plantlets to become wilted, right? This therefore tells you that it is very necessary for you to accustom the plants, to acclimatize your plants or to allow your plants to acclimate in the natural conditions through hardening and acclimatization, right? So how are we going to do this, right? So the culture containers, for example, you can actually start to proceed with transferring them to a greenhouse with loosened lids. This is to allow for at least some of the outside air to reach the plants, as well as for the plants to get used to the higher temperature. But why you put it in the greenhouse? Because the temperature in the greenhouse is more or less kept constant, right? And then you can also leave your plantlets in the shade for 3 to 6 days under diffuse natural light. Diffuse natural light means you have to put some sort of a netting to cover your plants, right? So that they can slowly adjust to the conditions of the new environment. And this in turn will help in semi-hardening the plants and will lead to subsequent shoot elongation. Like I've told you before, the leaves that are produced in vitro will be sort of non-functional when you transfer your plants to outside environment. So this will be replaced replaced by new leaves. Okay, this is why we need to do this hardening of the plants so that the plants can produce new leaves to support their growth outside of the in vitro environment. Okay. Why do we need to do this? This is because the transfer of your microshoots from in vitro to ex vitro condition, if you do that under direct sunlight, may cause photoinhibition as well as chlorophyll photobleaching, right? Where the chlorophyll pigments will be degraded, hence hampering with the photosynthesis efficiency. And then the in vitro to ex vitro transfer may also lead to transient decrease in photosynthetic parameters. Why transient decrease? Transient means something that's not permanent, a temporary decrease in photosynthetic parameters. Again, this is because there will be some adjustment that the plants will need to do, all right? Because they need to reduce the stomatal density, etc. So the amount of photosynthesis products they produce will be slightly lower compared to in vitro, right? And then previous studies have also suggested that photoinhibition may be the cause of their transient decrease in photosynthesis after transplantation. Photoinhibition is also due to the degradation of chlorophyll, right? Okay, second abiotic stress factor is humidity. So the retardation of the development of cuticle and epicuticular waxes as well as functional stomata under culture environment will cause high stomata and cuticular transpiration rate of the leaves when you take them out from the culture vessel, right? And then to avoid from this, the plantlets must be slowly transferred from high humidity to low humidity conditions. Again, hence why during the practical, you have to cover your plants with the plastic to allow for the plants to adjust their exposure to humidity. And the microshoots must be kept in the shade with the plugs or the culture lids loosened, and then after about 1 to 2 weeks, then they can be transferred to pots that contain sterile soil, the soil which has been autoclaved, and then sand mixture which are covered with poly bags, or in your case previously you don't use poly bags to cover but you use the plastic sheets to cover, right? And then the stomata and cuticular transpiration rates will gradually decrease due to stomata regulation of water loss, right, which has become more effective. And then the cuticle and epicuticular waxes slowly develop with time so that it gets thicker and thicker to prevent from excessive dehydration or transpiration. Okay, so if you don't do gradual hardening, without gradual hardening, even if the water potential of the substrate is higher than the water potential of the media with saccharides, this is your tissue culture media, right? The plantlets may quickly wilt because of water loss if this water loss due to transpiration is not restricted. And water supply can also be limiting because of poor conductivity of roots and root stem connection, which means that even though your tissue culture plants have successfully developed roots, the roots are not extensive, that's one thing. Secondly, poor conductivity, poor connection from shoot to the root system. So because of this, many plantlets will die during this acclimatization period if you do not subject your plants to gradual hardening. Okay, moving on to the third abiotic stress factor, which is carbohydrate concentration. Okay, so in the culture vessels, the plantlets that are developed are actually grown on a media which contains adequate or enough sugar, enough nutrients that allow the plants to follow a heterotrophic growth. Okay, heterotrophic means, have you guys heard of the term autotroph and heterotroph before?
Yes.
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 So what is heterotroph? What is heterotroph? Do you remember? Okay, we have two things, okay, two type of growth, autotroph and heterotroph. Autotroph are organisms, for example, plants that can produce their own food, okay, that can produce their own sugar for their growth. Heterotroph means organisms that cannot produce their own sugar, but instead must depend on other factors or other organisms to support their growth. So when you are doing plant tissue culture, your plants are actually following heterotrophic growth because you are actually supplying these plants with a lot of sugar, with a lot of nutrients. So they don't really have the need to conduct high photosynthesis because the aim of photosynthesis is to produce sugar, right? But because you are growing them on a media that has adequate amounts of sugar and nutrient, so they don't really need to conduct this very high efficient photosynthesis, so they can follow heterotrophic growth, right? And then studies have suggested that carbohydrate concentration will influence acclimatization process because during this process, plants will switch from heterotrophic to autotrophic growth. And any treatment before and after ex vitro transfer will increases the photosynthetic efficiency or photosynthetic capacity of the plants will improve subsequent plant establishment, meaning that you make it easy for the plants, all right, to transition from in vitro to ex vitro by supplying these plants gradually with what they require before you can transfer them to actual outside environment. For example, you can consider adding about 40 gram per liter sucrose prior to transferring the plants, for example, watercress, into in vivo conditions. And this has been shown to increase the dry weight of the established watercress plantlets. Right? And then in Bauhinia vahli, for example, rooted microshoots of this species, if you precondition them in different sucrose solution before transferring them in the potting mixture, was found to not enhance the percentage of survival, but will produce better quality shoots. Right? Okay, moving on to the fourth factor, which is ABA or abscisic acid, as well as AA in how plants tolerate abiotic stresses, right? So abscisic acid, like what we've learned before, is a growth inhibitor in plants. So abscisic acid is a critical compound or critical hormone for plant growth and development because it plays an important role in plant water balance and how plants can adapt to stress environments, including low temperature. Because it is a growth inhibitor, it also serves as a mediator or in order for plants to be accustomed to stress conditions. Okay, and abscisic acid is transported through xylem to the shoot where it helps to regulate water transpiration, which will cause water loss as well as will influence the growth of the leaves. And various stresses have been reported to induce the synthesis of abscisic acid. Therefore, abscisic acid is considered as a plant stress hormone. Right? And abscisic acid is a very important signal for plant resistance to pathogens, which will affect the biosynthesis of jasmonic acid and the activation of plant defenses in Arabidopsis. Right? For example, in Tagetes erecta, endogenous levels of ABA will help to regulate abiotic stress by controlling water loss from leaves, therefore helping plants to survive as well as to aid the growth of the microshoots when you transfer these plants directly to the field. Okay, again, like I've mentioned, ABA is a growth inhibitor or a stress hormone, but at adequate amounts of uh, production, they will not cause the plants to die, but instead they will help plants to sense that they are being stressed. Therefore, they will adjust the physiological processes accordingly, right? And then ascorbic acid will also help in abiotic tolerance. For example, abscisic acid that was expressed in transgenic potato through overexpression of glowase gene, okay, will cause abscisic acid accumulation and increase the tolerance of these plants to abiotic stresses. This is based on a study conducted in 2010. Okay, moving on to biotic stresses. Okay, example, exposure to soil microflora or soil microorganism. So another major cause why we observe high mortality rate of transfer from in vitro to ex vitro is because of the sudden exposure, especially the root systems to the microbial communities that are present in the soil. Okay, because these tissue culture produced plants, they do not possess sufficient resistance against the soil microflora, Why? This is because in tissue culture, the plants are actually grown under aseptic conditions. They are not exposed to any microorganisms at all. They are produced under a clean environment, right? So in the last few years, trials have been taken to expose young in vitro produced plantlets to useful microorganisms that promote the growth and encourage mutual association, so that plants will benefit from their association with these microorganisms as well. Example, endophyte. So fungi and bacteria which invade or live inside the tissue of plants without causing any diseases or injury to the plants. And in turn, they will also help to promote the growth of the host plants and the formation of the secondary metabolites related to plant defense, because these plants have sense that they are fungi or bacteria attached or inside their system, so they will increase the production of secondary metabolites as sort of a defense mechanism, right? And these secondary metabolites will also help in uh, allowing the plants to compete or to defend themselves from the soil microorganisms that are present in the field. And this technique is called as biohardening and has since gained a lot of attention. Right? For example, in vitro co-culture, you can actually co-culture your explants with bacteria or vesicular arbuscular mycorrhiza, which will induce developmental and metabolic changes in the derived plantlets, in the subsequent plantlets that you will produce. Therefore, these plantlets will have increased tolerance to abiotic and biotic stresses. And the induced resistance response caused by the inoculants caused by this bacteria or mycorrhiza are referred as biotization. For example, in 2010, Howe et al. Reported that the treatment of suspension cells of Ginkgo biloba with fungal endophytes have resulted in accumulation of flavonoids. There are increased abscisic acid production as well as activation of PAL, phenylalanine ammonia lyase, which are important during synthesis of flavonoids and anthocyanins. Okay, mycorrhization of tissue culture plantlets also have various advantages. For example, they can improve nutrient availability, soil pH, aeration, as well as protect the plants from water stress. For example, in a study done in 2008 on chlorophytum, it was found that more than 95% of the plantlets were successfully established in the soil after treatments with various bioinoculants, right? And then even the unrooted shoots also showed in vivo rooting when treated with several mycorrhiza. Unrooted means they actually transfer shoots that have not produced any roots directly into soil, and then these plantlets will produce root sections in vivo after transfer, right? Okay, so to summarize, due to the high mortality rate, plantlets must be slowly acclimatized to ex vitro conditions with high light intensity and low humidity conditions. And in vitro plants that are deficient, that do not have enough abscisic acid, but the content will increase during stress conditions, and this will help in acclimatization as well, because in this case ABA will sort of play a role as a stress indicator or stress mediator in the plants, right? And then carbohydrate concentration also play a role in acclimatization, as the plantlets are transferred from heterotrophic condition to autotrophic condition. Biotization of the micropropagated plantlets will cause enhanced growth and survival during lab-to-land transfer. And then plant endophytic bacteria and these VAM fungi also promote plant growth, resistance to pests, and increased productivity after transfer. Therefore, current research mainly focus on how uh, we can understand the plant microbe interactions, so that we can optimize this beneficial plant endophyte bacteria relationships, so that the plants can actually um, withstand the exposure to soil microflora, but at the same time also increases yield and increases the chance to survive, as well as to support their subsequent growth after transfer. Okay, so these are the references that have been used in the lecture today. Anybody has any question?
No, doctor.
Doktor cakap nama doktor. Let me see. Okay, tak nampak. Okay, somebody has a question from Faiz. If you autoclave the soil, doesn't it also will kill the useful micro? Yes, it will kill this useful microorganism in the soil as well. Right? Why do we do this? Like I've mentioned before, in culture, the plants are not exposed to any microbes at all. Okay, so that's why when you are doing acclimatization, you transfer them into a clean soil in an aseptic soil, because we don't want the plants to sort of like suddenly be exposed to that high amount of microorganism. So that's why in some research they are slowly treating the plants with useful microorganisms first and then transfer them to the field so that they have higher chance to compete with all of these microorganisms. Anybody else? Tak ada. If don't have, then that's it for today. So again, remember during lab session this Friday, you don't really have a practical, but we will use that period to do your test, which will cover everything that we've learned so far from topic 1 to topic 7, right? And then that test will be conducted in about 50 minutes. So after the test is completed, you can use the time to observe your cultures if you still need more data for your report, right? Everybody okay?
Okay, doctor.
Okay, doctor.
Okay, doctor.
Thank you very much, guys. I will see you guys this Friday.
Thank you, doctor.
Thank you,
Doctor.
doctor.
All right, bye. Assalamualaikum.